Hi everyone. In this video, we will discuss dissolution for poorly soluble BCS Class II and Class IV drugs. The BCS or Biopharmaceutical Classification System classifies drugs by solubility and permeability. Today's drug pipeline is dominated by Class II and Class IV drugs. These drugs have poor solubility, creating major challenges for dissolution testing, formulation design, and regulatory approvals. That's why robust biorelevant and discriminatory methods are more important than ever. For BCS class 2 drugs, solubility is low but permeability is high, so dissolution controls absorption. For BCS class 4 drugs, both dissolution and permeability limits absorption. That means slow dissolution is the rate limiting step. These drugs are highly sensitive to hydrodynamics, pH, and media composition, so regulatory scrutiny is high, especially when using surfactants or biorelevant media. Method justification is critical. According to Noyes-Whitney and Nust-Brunner, dissolution rate depends on surface area, solubility, and agitation. For poorly soluble drugs, all three are reduced. Hydrophobic surfaces resist batting. Solubility does not increase much across pH. The diffusion boundary layer becomes thicker and particles tend to agglomerate instead of dispersing. That's why low solubility drugs dissolve so slowly. BCS class 2 and class 4 drugs often need advanced formulation strategies like nanosizing, solid dispersions, or lipid systems. Your dissolution method must match these chosen strategies. Always consider pK and pH dependent solubility. Solubility screening from pH 1 to 8, particle size distribution, which is critical. Polymorph stability and log P or hydrophobicity. Method design must be science driven. Surfactants forms misleads that improve wetting and drug solubilization, but too much surfactant can reduce discrimination between formulations. So use minimum needed, typically 0.1 to 1% SLS. Biorelevant media mimic food effects and intestinal solubilization, like FASSIF and FESSIF or SGF and SIF with wild salt and lecithin. Always adjust pH to match the drug's ionization profile and avoid media that are too strong. They can hide real differences. USP apparatus 2 is the most common starting point in QC labs. If particles float or agglomerate, consider USP 1. For low solubility drugs, USP4 is ideal. It provides continuous medium flow, creating near infinite sink conditions and better hydrodynamic control. USP4 improves biorelevance, but for routine QC, USP2 remains more practical, often with pH adjustment or mild surfactants. Surface active agents reduced interfacial tension, improving wetting, but overuse creating non-discriminatory methods. So add only minimal wetting agents like SLS or polysorbates. Always degas media to prevent practical floating if justified. Increase agitation 50 to 75 rpm. Use sinkers for floating capsules or tablets and reduce aggregation with gentle pre-wetting. Balance is key for meaningful dissolution testing. In USP4 apparatus, continuous flow follows Henry law. Dissolved drug is constantly removed, maintaining sink conditions. Regulators closely scrutinize co-solvent use because it can overestimate dissolution. So first, increase medium volume up to 1500 ml if justified. In USP4, you can also increase flow rate, use surfactants or co-solvents sparingly, and strengthen buffering to maintain stable pH. scientific justification is essential. Regulators expect discriminatory dissolution methods. A good method must detect 20 to 30% change in particle size distribution. Why? Because dissolution rate is proportional to specific surface area. Small PSD shift cause large changes in dissolution profile. So, PSD must be tightly controlled during manufacturing, and your dissolution method must be sensitive enough to detect these changes. That's two methods discrimination. For many BCS class II drugs, food dramatically increase solubilization. That's why testing in biorelevant media is so important. For use FASSIF and FESSIF, they mimic bile salt and lecithin action. This helps predict fed versus fasted behavior in patients. It also supports IVC model development. Biorelevant testing leads to better clinical predictability. Regulators closely review any use of surfactant or strong media. Why? Because the risk of non-discriminatory method is high. For BCS class 4 drugs, both solubility and permeability are low. So dissolution may stay slow even with surfactants. Stronger media might be needed, but they can hide formulation problems. So, focus on proper batting, clear surfactant justification, and strong discriminatory power. That's what regulators expect. Regulators require proof of discriminatory ability. If your method is too strong, especially with excess surfactants, it hides formulation differences. So, test intentionally buried batches, different PSD, binder and lubricant level, wet versus dry granulation, and polymer content in solid dispersions. Avoid over-aggressive media and ensure robust hydrodynamics. A good method reveals changes, it never masks them. Most modern APIs fall into BCS Class II and Class IV. So, systematic approach is critical for regulatory success and predictable in vivo performance. Start by understanding API properties like PK, PSD, and polymorphs. Choose balanced media, right pH, minimal surfactants, and biorelevant options when needed. Select the right apparatus USP2 or 4, improve batting, and maintain sync conditions. The PSD sensitivity to confirm discrimination and avoid over-aggressive media that hide the real difference. That's the formula for robust dissolution method. Thank you for watching. In next video, we will discuss alcohol-induced dose dumping studies, why and how. Please subscribe us for getting update on upcoming industry application-oriented videos, and stay tuned because in this 62 video series, we will cover dissolution method development, validation, troubleshooting, and many real industry examples. Please share your queries in comment section. See you in next video. Thank you.